moved offshore, they'll recruit back in eventually to coastal habitats like the Indian River Lagoon. And this is a little time lapse of what we do. We go out twice a month. We are in several little boats. We set a net, it's a tangle net, and it's kind of like a mist net, I guess, if you're trying to capture and tag birds. We set this long net, it's about 455, is it meters or feet, Bill? Meters, thank you. And we end up catching, over the course of 3 hours of sampling, upwards of a handful of turtles in the net. Sometimes we'll catch some rays, sometimes we catch a lot of rays that we release. We'll also catch some sharks as well that we release. But if we see, what we have is along that net we'll have a whole bunch of buoys at the top that are floating. And if a turtle or something gets caught in it, those buoys stand up and they start to dance around. So that's an indicator that we need to go check out the net in that area. We sneak up on that area, we might hand over hand pull ourselves up to that dancing buoy, and then we use a dip net to get the turtle, bring it on board the boat, disentangle it, and then we take it to a workup boat where we will science the heck out of them. We will end up taking measurements, similar to what we do with the turtles on the nesting beach. We'll take tissue samples, we'll take those biopsies for stable isotope analyses. We're also looking at the genetics of these guys because we are getting turtles in the Indian River Lagoon that are coming from a lot of different places, including Mexico. So it's a mixed stock developmental habitat, meaning that turtles that hatched out on nesting beaches in the region, not necessarily just in Florida, go to the Indian River Lagoon to grow up. And we will weigh them. We can actually weigh them because they're small enough for us to do it without breaking our backs. We don't do that with the nesters on the beach. But we'll take blood from them as well. And we're doing some really neat work with Dr. Anna Savage, who is an assistant professor in the biology department where I'm working. We've formed a really fun collaboration. She's a disease ecologist. She works with frogs and some gopher tortoises, and now she's working with us on a tumor disease that you see on about half of the turtles that we capture in the Indian River Lagoon. It's called fibropapillomatosis, otherwise known as FP. And what happens is we think it's associated with a virus, likely a herpes virus, a turtle herpes virus. And at some point, typically the smaller turtles, the younger turtles, we found will develop these tumors that they're very warty looking. They might look a little bit like cauliflower or broccoli. And they'll grow these tumors for a period of time, but then those tumors can also disappear. They can regress. It becomes a problem if those tumors cover a turtle's eyes or block their throat, or it becomes kind of systemic and attaches internally to a lot of their organs. But any turtle that is heavily covered in these, where they're clearly impaired in movement or foraging behavior, we will send them to a local rehab facility. Otherwise, what we'll do is we'll take samples. We'll take blood samples, we'll take tumor samples. We're looking at the presence or absence of the virus. We're trying to look at different mechanisms that might be associated with the development of those tumors and the development of the disease or the regression of the tumors. We're also starting to do some really neat gene expression work as well. So if you need another person to give a talk at some point, I highly recommend Dr. Anna Savage. She can talk all about disease ecology in frogs and amphibians and also sea turtles. We do mostly catch green turtles, about 150 green turtles on average every year. Half of those have FP, those tumors. And we do catch mostly green turtles. We'll catch about 40 or 50 loggerheads every year as well. And then very rarely we might catch a leatherback. I think only 2 have ever been encountered over the years. And rarely a Kemp's Ridley, which is in the lower right-hand corner there. Those Kemp's Ridleys are fairly rare. They stick mostly to the Gulf of Mexico. So all of our coastal work contributes to a lot of different directed research projects. We're looking at genetics, we're satellite tagging and tracking some of the turtles both on the nesting beach and hopefully soon in the lagoon. And so we look at their in-water movements, their behavior, where they're going, where they're migrating. We also, as I mentioned, take blood and tissue to look at genetics, stock of origin, and some of the disease ecology questions. And we're also looking at the diet of those animals. What are they eating? And it's a really fun day on the boat when one of them poops, because we'll collect everything. We get excited. But we've been doing some lavage, which is just kind of cleaning out the esophagus of the animal, collecting what comes out, and trying to determine what they're eating very immediately, like what they've recently been eating. And we're also starting to look at the microbiome as well, taking cloacal swabs, basically swabbing their rear ends and finding out what the microbiome looks like. Now, the biggest portion of our, the biggest data gap, or the biggest data gaps that we have with regards to sea turtle life history and understanding what these little guys are doing is really associated with the offshore years, those early years from the time they leave the nesting beach as little hatchlings until they return to places like the Indian River Lagoon as larger teenagers or juveniles. And those little guys, I mean, they just disappear from our radar. And it's really, one of my favorite things in my job is introducing students to hatchlings. There's something about hatchlings that just people find joy with. And almost everybody that I encounter that hears that I work with sea turtles, almost everybody has a story. They'll have seen a turtle swimming somewhere, say in Hawaii when they were on vacation. They may have seen a sea turtle nesting. They may have been on the beach when hatchlings emerge. And it's really the hatchlings that kind of grab people. Baby turtles are incredible science STEM ambassadors. And so one of our favorite things to do is try to catch pictures of our interns when they see their very first hatchling. And the middle video is some little baby leatherbacks that were just emerging from a net, or from a nest. And that first image is one of my graduate students who happened to be out doing nest inventories on a day when he had 3 different species of hatchlings that he found, which was exciting. So what we, all of this is Jessica Den from this past summer. She saw her first baby hatchling, and it was, I think, kind of life-altering for her. She was really excited. So what we do is we will subsample some nests, we'll mark them, and then after about 45 days of incubation, 55 days or so, 45 to 55 days, we'll start to monitor those nests for hatching, signs of hatching. And the signs that we look for are in that first image. It looks like somebody took a cereal bowl, pressed it down into the sand, so there's a bit of a depression. And then you see tiny little tracks that should be hopefully heading towards the water. And as these little guys emerge up through the sand, poke their little noses out, and then they start crawling down the beach, hopefully they're going to be orienting to light that's reflecting off of the ocean and not condos that are lit behind them. Hopefully it's a nice dark beach. The turtles do get distracted. They'll get disoriented by strong lights on the beach, which is why we have local ordinances that reduce beachfront lighting. And those little babies will orient to get into the water. They then orient to swim into the waves. And then they, we think, kind of click in with this innate compass map, this innate sense of where in the world they are based on the Earth's magnetic field, very similar to a lot of different bird species. And at that point, they swim like crazy for about 24 to 48 hours. They're using up the extra energy that they have absorbed from the egg yolk that they've grown around. And then they get offshore and then they slow down. And hopefully by then they'll have encountered a floating mass of algae known as sargassum. And that, at that point, from the time that they kind of get through that frenzy period to the point that they get offshore, that's from that point on until they return back to coastal habitats, that's known as the sea turtle lost years. And again, we don't know that much about how long it takes for them to get out there, how they interact with that environment out there, how they're swimming through things. Are they swimming with currents, against currents? Are they swimming? One of the kind of the biggest questions about these guys is whether they're passive little drifters or active swimmers. We know some about these little guys. We know that they do swim like crazy, they get offshore. We know that they associate with this brown floating algae known as sargassum. It sometimes washes up on our coastal areas. It's been a problem down in the Caribbean with big piles of it in the last year. We know that they live at the sea surface. They have tiny little lungs at this age. They're not going to be diving 300 meters to find food because they just don't have the lung capacity for it. But they mostly stay at the sea surface. We know that they are offshore, meaning that they're off the continental shelf. They're in waters that are deeper than 200 meters. The continental shelf is in that first picture. It's that turquoise colored, the bright green is land. The darker blue is the oceanic, the offshore habitat. And they're known to associate with that sargassum. The sargassum rafts up. If you do any offshore fishing, you might target rip lines. You might target areas where the sargassum kind of gets pushed together, and a lot of different little creatures are found there. Little crabs and crustaceans. You have birds that might follow some of those rack lines. You have baby turtles that are known to associate with those habitats. And it's thought that they get a little bit of protection from predators that might be circling around, hungry, looking up, looking for them. And then it's always been assumed, at least until recently, that these little guys were passive drifters. They'd get offshore, burn off that little extra energy during their frenzy period, and then passively drift with the ocean currents in a big circle around the Atlantic, at least for turtles that emerge off of Florida's beaches. But the question is, they're built to swim, why wouldn't they? One of the other aspects, and this is kind of where encounter bias comes in, we know that they're tiny little hatchlings when they leave our nesting beaches. They've been observed off of the Azores by people that fly to the Azores and get in boats and go a short distance around the Azores, that they're maybe about the size of a cereal bowl, maybe a dinner plate off of the Azores. This is for loggerheads in particular. By the time you get to the Canary Islands, they might be a little bit bigger, 40 centimeters or so, maybe dinner plate size to turkey platter size. And then by Cape Verde, they might be a little bit bigger. And then by the time they come back into our coastal habitats, they're about the size of the backs of your chairs maybe, or a little bit bigger. So we've assumed, based on finding these turtles in places that we can easily access, that hey, maybe they just kind of take a ride around the Atlantic in those currents that form what's called the North Atlantic Subtropical Gyre. And if you're familiar with off the coast of Florida, there's the Gulf Stream that rides up the coast of the east coast of the US. That turns and heads over towards Europe, and it becomes the North Atlantic current. By the time it gets to the Azores and heads south, it's the Canary current, and then it's the the North Equatorial current, which is just above the equator, and it brings everything back towards our neck of the woods in the Caribbean and eventually up into Florida waters, and it turns back into the Gulf Stream. So the idea is that, oh hey, we've caught turtles in these areas. They're different sizes. It makes sense. It takes time to get from point A to B to C to D, and those turtles have been examined genetically to know that they likely originated from Florida beaches. They have the same genetic signature because, if you recall, turtles have very strong natal phylopatry, site fidelity, to their originating beaches. So those genetics tend to create a signature over time in those areas. But nobody's really focused on what's going on in the middle, in the Sargasso Sea. It's not really been very well studied, in part because there's not a landmass there for us to travel to and then take a boat from. A really cool study was done by Dr. Ken Lohman up at the University of North Carolina, Chapel Hill. He took baby turtles that had never been in the wild before. They hatched out, he put them in a tank, he put them in little bathing suits, and then he attached a tether to them. And around this tank that was a circular tank, he attached them to a little tether that was attached to a computer that could tell which direction the turtles were going in a 360-degree circle. And he created coils around the tank where he could then replicate the Earth's magnetic field in different points along the gyre, this big circular current system. And he found that by replicating the Earth's magnetic field, say, off of the Carolinas, or that first North Florida, I guess, all the turtles were orienting to the east-northeast, or maybe a little bit to the south. In the area off of the Azores, those turtles would orient to the south. And then in the area that was just past the Cape Verde and Canary Islands, they would be orienting to come back our way to the Florida area. And so that's really neat. That's how he showed that these turtles had that magnetic sense and that innate compass, because these turtles had never been in the wild before.